Welcome to the new episode of the webinar series hosted by the EASD ECA. I am Konstantinos Toulis, endocrinology and diabetes physician based in Thessaloniki, and my co-host today, Dr. Caterina Conte, assistant professor in diabetes and general medicine based in Milan. Let me introduce Professor Krishnarajah, who is a health data science and public health professor at the University of Birmingham, UK. He's also joint director for the Center of Health Data and Science and deputy director for the Institute of Applied Health Research at the University of Birmingham. He and his team have been very active in producing high-quality research on diabetes, pharmacoepidemiology, diabetes complications, and gestational diabetes. And he is particularly interested in automated methodological approaches for clinical epidemiology research. And I need to thank him for accepting our request to give his insight on real-world evidence. So, Chris, welcome. It's an honor for us to be joining our initiatives here. Now, we may follow a more vivid approach today, take advantage of the expertise here, and we're going to talk about the applications, challenges, and perspectives of real-world evidence in diabetes research and the importance of health informatics in diabetes epidemiology and public health. So, Chris, we have all witnessed and observed a trend towards incorporating more real-world evidence in diabetes literature. And since you have been very active on this front, we wish to know more details about this real-world evidence in diabetes. So, what is real-world evidence in diabetes? Where do the data come from? Are they readily available to anyone? What data sources are commonly used in real-world evidence in diabetes? Could you please give a short overview?
Thank you, Dr. Konstantinos and Dr. Katerina. And welcome, everyone. I'm Krishnarajah. You already heard the introduction of myself. So I'll directly go to the question in terms of where real-world evidence sits within diabetes research and what do we mean by real-world evidence. So terminologies can be quite confusing. Sometimes you hear electronic health record data, sometimes you hear administrative data, sometimes you hear real-world evidence data. I will primarily focus on electronic health record data because real-world data could also mean some of the pragmatic trial data we do in clinical practice. So where does real-world evidence data come into play when it comes to diabetes research? So naturally, clinical trials are the gold standard for any effectiveness study, and no one can dispute that particular argument. So real-world evidence data comes in various time points when you're looking at effectiveness study. One, obviously, it comes when it comes to drug safety. So you all know some of the drugs that had long-term effects that were later discovered in diabetes and had to be discontinued. And the reason being most of the clinical trials are short-lived. And therefore, for longer follow-up and rarer outcomes, it is only possible to find them through observational data, and real-world data comes into play there. So drug safety is a huge area in terms of real-world evidence. Secondly, real-world evidence data is also useful where a drug effectiveness has been proven, but it was on a population that are very much restricted to a typical trial population, people who have diabetes, don't have comorbidities, often not older adults, and often not in pregnancy. So this means you need to know the drugs that have been proven effective, like SGLT2 and GLP-1 agonists, do they work in people who were not part of the trial population. So these are usually older adults, or it is usually people with multiple long-term conditions who are often excluded from trial setting. So you can be confident as a clinician when you prescribe these medications are also prescribed to population that were not tested. So that's a second opportunity. A third opportunity is where you have performed a trial but have excluded a particular subgroup. For example, the initial cardiovascular disease trials using SGLT2 were performed in high-risk cardiovascular disease population. And what you don't know is, does it have a benefit in low-risk cardiovascular disease population? And one of the first publications, Dr. Konstantinos and we published, showed that actually it does work in low-risk population. Okay, so that's another third application. A fourth application is around identifying potential additional benefits that may not have been observed in trials. Just as you have recognized SGLT2 now used in cardiovascular disease, SGLT2 is now used in chronic kidney disease, and it won't be long before we will be using it for other purposes. And these can be tested before you do a trial, so that you don't waste volumes of money before going and doing a trial on a medication that already exists. So we call it repurposing medication and trial emulation ahead of doing new studies. The other element is also around planning clinical trials. So, for example, recently we have been doing a trial around atrial fibrillation and anticoagulating low-risk population. But before doing a trial, you need to know if this is a public health problem. Do low-risk atrial fibrillation patients are at high risk of stroke? Secondly, you need to know what is the incidence of the outcome of interest stroke, so that you can do a proper power calculation before you go and do a trial. Thirdly, if you are going to actually do an electronic health record driven clinical trial, in which case where you recruit patients from electronic health record, then you need to know what number of patients you can expect to get. And finally, you can also do some trial emulation, because it's a medication that already exists, to understand if this is likely to work in a population. And I have just given you examples where it involves a medication. I can give you more examples which does not involve medication and where real-world evidence can be of benefit. I'll stop there, Kostas, and I'll take other questions before going on to any other answers.
Okay, thank you. But Chris, so you have provided the summary of how real-world data can expand, extend the findings of traditional RCTs. In terms of planning and selecting the population, in terms of validity of the studies, in terms of levels of evidence, and in terms of other type of advances, such as cost or speed, or is there anything that we need to know more about this novel methodological approach?
Definitely, I always say when you want to do real-world evidence, there are three key things you need to be aware of. One, the data. You need to understand the data really well. Otherwise, you may end up interpreting many findings incorrectly. Second, you need to have the right tools to process the data. So if you don't have the right methods and tools, again, you can end up with biased results. Third, you need the right people in terms of doing the research, because real-world evidence is multidisciplinary. You need the clinician, you need the data scientist, you need the computer scientist, you need the epidemiologist, you need the statistician, and of course, the patients themselves in shaping your research question. So without those three key things, knowledge of the data, the right data at the right time, 2, the technologies, and 3, the people, it is quite confusing, can take very long time, can feel a huge burden. But if you have those in place, then you can do high quality real world evidence research.
Is there any advantages in the processing of data in the software that can support this type of studies? Is there any quick example you may show us? Would you be kind enough to show us the main methodology software support that you have developed?
Very happy to share some slides, and if time permits, give a demonstration as well. But before that, can I get a feel for how many minutes do I have to do so?
We have plenty of time. All that is needed is available.
Okay, so I'll share some slides so that it's a lot more clearer. I hope you can see my screen.
Mm-hmm.
And something called Dexter at the top. Yeah.
Okay.
Okay, I'm going to quickly run through a few slides I have prepared for an Alan Turing Institute presentation. So if you would like to hear more about it, we can send you all a link, which has got a longer, close to 45 minutes talk. The good news is I won't go that far on this occasion, so it's going to be short. So I'm going to talk to you about a technology we developed over the last 7 years, which interestingly was a time when I met Dr. Konstantinos, and who was part of the team who helped us to develop this technology. And I'll take you through that and how that has revolutionized the way we do research in University of Birmingham and definitely globally as well. So it's a semi-auto platform to do electronic health record research. And we here in University of Birmingham are fortunate to have access to a large number of databases. The common one we use is CPRD Gold and Aurum these days. Previously, we used to use a database called TIN. So CPRD is a primary care database. It has close to 30 to 40 million patient records. So you can imagine how many diabetes patients will be there in that sort of population. And it has electronic health record data. So it has all the prescription diagnosis, observations, blood results, so much wealth. So you could spend all your life just doing research on that database. It is a costly database, so we do pay around £130,000, but we do recover it through grants and various other pots of money. We also have access to secondary care database like Pioneer, which is a hospital database, but they are restricted access, and also pregnancy database. So this is THIN database, which has 18 million patients, and you can see what sort of data is there, therapy, medical records, and so on. And when we extract data from this for a particular study design, so many of you are junior early career researchers. So when you want to do a pharmacoepidemiology study, even if you look at my study, it has evolved to better methods over time. And these are very complex study designs, and the latest study designs we use are very complex. We call it prevalent new user design. And this involves really rigorous epidemiological methods that need to be applied. Otherwise, you will end up with a whole range of bias. But if I have a study design and a protocol, if I ask someone to extract that data for me, it is very costly because this person has to have lots of expertise and ability to do so. And if I ask 10 people to extract data, all 10 of them will give me 10 different data sets. Trust me, it is true. If I ask the same person to give it at 10 different time points across over 10 months, they will give me different data sets. So it's not terribly accurate or reproducible. So considering this used to take us 3 to 6 months before we get a database to analyze it. And I found that very frustrating. And I thought, if I can work with multidisciplinary team of data scientists and computer scientists, I can automate. Can I create the ChatGPT for healthcare data? So that was what we wanted to do. And this is what, when people say electronic health record data, they say it's dirty. And I always compare it with crude oil. So those who know crude oil, we use crude oil. We do what we call fractional distillation. And when you do fractional distillation, when you pass the crude oil through different level of temperatures, you get the less pure bitumen at the end, which you use to make roads, but you also have petrol, quite high quality to drive cars, but you have kerosene and so on, and these things do amazing things. They fly planes and all sort of things, okay? So if you know how to handle this data, electronic health record or routinely collected data through the fractional distillation process, through processing it carefully, you can create amazing research and high quality papers. You need to understand the limitations, and you need to acknowledge, and you need to explain what the limitations are. As a public health physician, I always believe that sometimes you need knowledge sufficient for action, but insufficient to satisfy your intellect. So as an academic, I may not be satisfied, but as a public health physician, if that information helps me to make decision, I'm more than happy, because I'd rather make decision with intelligence rather than empirical decisions. So what we did was we tried to handle this problem. How do you extract data per study design? And what we did was we automated this program by engineering. And what we ended up with was where we can do studies like cohort studies, case control studies, cross-sectional studies, and now complex pharmacoepidemiology studies within short time scales. What used to take 6 months can be done in 6 days, sometimes even in 60 minutes. So that's the power of such technologies to generate new knowledge from existing data. And this means junior colleagues, lots of early career researchers are able to handle this data, handle research questions, and produce good, high quality research. So Krishna on the left, and you can see Konstantinos and Peter Tino. I think it was all European team who got together to do this project and deliver this, so that it is published. And all of a sudden, we have now published more than 100 real-world evidence papers in the last 3 to 4 years alone. And you can see that you can do things at scale as opposed to previously. As I told you, we have extended this to pharmacoepidemiology and other baby data. And here's a typical simple study around polycystic ovarian syndrome and risk of non-alcoholic fatty liver disease. And the beauty is it showed, not that it showed they have twice the risk, but it also showed that this risk was independent of the BMI. So it doesn't matter if the PCOS woman had a normal BMI, they were still at high risk. What it also enabled us to do is take 70,000 testosterone values and look at the risk of polycystic ovarian syndrome. And what we found was where high testosterone levels were there, the risk of NAFL was high. And a similar paper in diabetes we published, where we showed high levels of testosterone was linearly associated with both women and men. So higher the testosterone, greater the diabetes in women, lower the testosterone, greater the risk of diabetes in men. So what we hypothesized here was polycystic ovarian syndrome and their risk for diabetes and non-alcoholic fatty liver disease is not just driven by BMI, but also by the high androgen levels. And this meant we were able to, one of our colleagues whom you all probably know, Professor Vip Kayal, went on to get a large grant to develop anti-androgen property medications to tackle this problem. This is what we call informatics consult. Just imagine you are on a ward round and you have a question and you want to answer within a few minutes. So here is a question we demonstrated. So in Birmingham, in wave 2, we wanted to know if you have diabetes, what prognostically, what impact it has if you were to be admitted with COVID. So usually severe patients receive dexamethasone, so we kept dexamethasone treatment as a proxy outcome for severity of disease, and also looked at mortality. So when we looked at our hospital data, there were 518 patients who had COVID and diabetes, 1,400 patients who had COVID but no diabetes. And you can see their ages are different. Diabetes patients were older, so naturally you will expect worse outcomes in them. But after propensity score matching, not just the age, but also another 20, 30 parameters like serum albumin to various other factors, CRP and so on. You can see the ages are similar and you can get results. And what results did we get? We got a hazard ratio here where you can see people with diabetes were more likely to receive dexamethasone, so suggesting they do get severe disease. But in the wave 2 of COVID, unlike wave 1, we did not observe increased mortality. This is a reflection of better care for diabetes patients during the second wave. You all may recall in the first wave, we didn't know how to manage diabetes in COVID patients. Blood sugars were all over the place. And what we learned from wave 1 and better treatment guidelines, the mortality were okay. But the staggering thing is we performed this research in 3 minutes, okay? And I'm not making this up, okay? So, but it is a small database and you were able to do this type of research. What we have now got is a £2.5 million to do something more exciting using artificial intelligence. So imagine if you're a doctor and you don't know what is the next best second-line treatment for a given patient, because you have opportunities to give DPP-4 inhibitor, SGLT2, GLP-1 agonist. You don't know what is the next best treatment because your patient who is sitting in front of you is not a typical diabetes patient. This patient has Addison's disease, has depression and rheumatoid arthritis, okay? And the GP, the trial evidence doesn't actually lend into these sort of patients. So imagine if we have artificial intelligence which can learn from data and give patient-specific results to say how the choice of medication can help with diabetes, but also help with the coexisting comorbidities. And 2, also help with the predicted comorbidities in the future. So if they are predicted to have, for example, dementia, would you choose GLP-1 agonist as opposed to SGLT2? I'm making this up, but that is the sort of question we are starting to answer. Beyond that, observational studies, and informatics consult, we recognize we can actually use Dexter for clinical trials. So we have now got a trial on gestational diabetes where eligible population are recruited through Dexter, passed on to the GP practices, and then the data, the outcome are measured on the data we collect. I will stop there, and if we can always show a demonstration, but I'm conscious time may be a constraint. So yeah, very happy to take some questions.
Okay.
Thank you. It was actually enlightening because it's very complex. So how do we recognize a good study? Is there any, because maybe we read a paper, so are there, I don't know, some points that we should check to make sure the quality of the research is good?
It is very difficult, and I acknowledge this issue because, for example, if you take SGLT2, there is a range of varying real-world evidence studies, including our own studies, have become much better than the previous ones in terms of their effect estimates. Okay, so first principle, and I presume most of the people here are clinicians. Am I right in saying that? Yeah. So most of you would have got training in evidence-based medicine, and that means you know how to critically appraise studies. And you should know how to appraise a clinical trial as much as a cohort study. But then when it comes to pharmacoepidemiology study, using real-world evidence, it becomes even more difficult because the biases are even more difficult to understand and elicit, like immortality time bias, prescription by indication bias, so survival bias. How do you right censoring bias? It's all like terminologies we never heard of for many early career researchers. So the key thing to look at if you're looking at a drug is, do they have a comparator drug? Is it an equivalent comparator? So quite often for SGLT2, they use DPP4. Is it appropriate? Or should we use something else? So even if you look at the best pharmacoepidemiologists, at times have changed their comparator drug coming from Montreal, for example, to different drugs in different studies. And how do they set up the index date, the date they start the follow-up? Is it on the date of prescription? Is it after 4 prescription? How do they put the lag period? These are very complex epidemiology. I might take the next whole year to explain. So I do share the concerns. But the key thing to look at is use your clinical intuitiveness to understand, does this make sense? So for example, when the latest study we are doing is around SGLT2 GLP-1 agonist and how they influence dementia outcomes. Okay, and there's already a few papers. Many claim large effect sizes, which we are not seeing. And how do we, so always look for the clinical knowledge as much as the methodological, but it is not an easy one, and always make sure you discuss in a forum rather than try to make sense out of it individually.
Mm-hmm. So, Chris, imagine you are a young diabetologist and you wish to do a PhD or further, so postdoc study. Is there any kind of prerequisite to have, like a type of background education you need to get grasp of what's going on in this field? Do you think that it needs a specific knowledge or something? For someone to go and work within a team of real-world evidence methodology group, does he or she need to have a specific background?
Yes, so early career researchers, obviously people do it in different ways. Some might get a fellowship for a PhD, some might get a fellowship to go and spend just a year abroad, or some might have to just do the research while doing many other things as well. So the first thing to understand is you will need to have a good background in epidemiology. That's the first thing I would say. You need to know statistics, but people can help you with the statistics. So usually real-world evidence teams will have statisticians along with them. I would always encourage people to look out for courses around this and start with basic epidemiology. Don't go into pharmacoepidemiology because they are complex and it will take time to get into it. Look for places that have tools like what I showed, Dexter. Otherwise, you might go and ask for a question and get the data only in 18 months' time. So just make sure you choose the right place to go and work. When you make sure there is a multidisciplinary team, you're not left alone with some videos to learn courses. And it's you need to be around with people, not on Zoom. So keep those things in mind and choose the right place to go to. And it is very difficult because naturally we are fortunate. We are in a position to pay for this database. We were not able to do so previously. It's very expensive. So there will be a cost element. And UK, there is quite a few places. It may not exist in every country. So you need to think around that. For example, Cegedim provides European data, and we are fortunate to get that data from next month onwards, where we will have French data and data from elsewhere, Italy and so on, but smaller scale data. So yeah, be on the lookout for those sort of places and opportunities when you choose to have your placements and so on.
Cool. Chris, there is a couple of questions coming from audience. Would you like me to proceed with them?
Please do.
So there is a colleague asking about concerns with data quality. So how is the integration of databases from different health systems done? So in fact, I believe that he's asking about how do the results from different databases compare? Have you noticed any pattern that might be worrisome or any kind of inference driven by differences?
Very true, and a really good question. So data quality is always an issue, and it will always remain an issue for life. Okay, so that's the first thing.
Yeah.
So how we handle it is all of them have a purpose as far as you use it with the right skill and the right intelligence, derive the right intelligence from it. For example, if people don't code diabetes well, you can't find the population of interest, okay? But that is less of a problem here in UK because general practitioners are incentivized to build a diabetes register. Even then, there will be some misclassifications. But this might be a major problem in another part of the world, and you have to be aware of it. Second, there is exposures. So drug exposures are really good because they all get recorded in general practices. Whereas if it's some other exposure, if the disease itself an exposure and it is a rare health condition that is not well recorded, it may be an issue because selective populations might be recorded. But the key thing you need to ask is, are they differentially recorded? So if you have an exposed group and unexposed group, and if they're differentially recorded, then you create bias. If they're not differentially recorded, but still under-recorded, your effect size, as we call it, is unlikely to be affected. It's the true incidence rate that will be affected, and your confidence interval will be wide, which means you may get a null effect simply because the confidence interval crosses the null effect. So this is why I meant you really need to have the understanding of the data, understanding of the design through epidemiology, and understanding of the analysis through statistics to look at these things. So even if the data quality is poor, it depends why is it poor. So if the outcome are recorded less, but are equally recorded in both places, effect size-wise, it's okay. But if you start calculating measures like numbers needed to treat equivalent, it will be wrong because the absolute risk is wrong in the first place. So it's having all those knowledge you have to bring in when you interpret, and then naturally acknowledge all the limitation so people can have a balanced view when they interpret this data.
Well, an interesting question has come in, and I know that you have something interesting and relevant to say. So Dr. Arne is asking, that whether there is a plan to incorporate measures like genetic risk scores to Dexter databases. Is there any?
Yeah, so the first thing is to differentiate that Dexter is not a database. Dexter is a software that sits on database. Okay, so and it can obviously sit on any database with some configuration. So in answer to that question, yes, if you put it on biobank data, UK biobank data, then naturally, which has both electronic health record data almost for 50% of the patients, there is genetic information, SNPs and so on. So you can build what we call clinical data plus genetic risk scores. I'm not a huge fan of genetic risk scores because it's overstated than what it is, but it has its value in certain places. When we achieve more integration of genetic data along with clinical data, that's when we will see the best utilization of those scores.
Do you think that this may come soon, or it might take some time to develop that?
So in UK, of course, in Health Data Research UK, within Cambridge University, they do quite a bit of genetic risk scores by Professor John Danish's group, and they are hoping to link it with clinical data to make better sense out of it. They have published a couple of papers, if I remember correctly, one on cardiovascular risk around JAMA. But I need to, but how much will they do in clinical practice is a question. But it is all about, at this time point, their value is limited. But things can change very rapidly. These things can become easily accessible, highly connected to clinical data, and doesn't cost additional funds as such, like many things have happened over the last few years. And in that setting, the incremental benefit of these polygenetic or risk scores can have a huge role to play, but more likely to be in terms of treatment choice and precision medicine rather than disease prediction as such.
Okay. So there's another question from audience asking about whether real-world data and the connection with big data and the differentiation of these key concepts, are they similar? Are they different? There seems to be a confusion around the terminology, but you have already mentioned that it's not the key point here. The key point is not the terminology and the typology, but rather the essence. But
so I would say the key thing is there's no real definition for these things. So people say big data, real world data, routinely collected data, all electronic health record data, and so on. But the key thing you are looking at is large data sets that previously how we managed in traditional setting are becoming increasingly difficult. And we need those technologies and methods, advanced methods to do so. And those do exist now, and therefore are able to process this. Process in terms of computational power, but also methodology, but also the knowledge we have brought in. So I wouldn't worry too much about those wording of this data.
Let me get back to your previous example, like the example you have mentioned about the COVID, diabetes patients, and your rapid response. So you have showed some results, and you have mentioned about the type of handling, the propensity score matching. Would you please expand a few words? What is this? I stumble upon this term when I read a real-world evidence paper. What is propensity score matching, let's say, and why is it so important, and how does it compare with an RCT?
Compare with?
The randomization, the typical randomization, from RCT.
Yes, so when you do this technique, real world evidence, we call it trial emulation. So you want to do exactly what happens in a trial. Naturally, you're not randomizing here, but the whole purpose of randomization is to achieve equal balance between both groups. But in RCT, you achieve equal balance in measured confounders, things you have measured, but also unmeasured confounders. By using propensity score matching, all you are doing is trying to match measured confounders and assuming and hoping that unmeasured confounders also become equal. Okay. So what you are trying to do is create two populations that are similar. And you can avoid doing propensity score matching and just do simple regression analysis and do some adjustment. But when you have large number of variables, 20, 30, people even do 100, 150 variables to match, it's impossible to do adjustment through usual regression methods, and you use propensity score matching. So we do it to create two equal groups, so they are similar to RCT population. And in doing so, we try to avoid what we call prescription by indication bias and potentially also confounders. So that's the purpose. But let's not go into detail because propensity score methods itself, there are 4 or 5 different methods. So you will all of a sudden come across inverse probability weighting to stratified propensity score matching to all sort of things, but I wouldn't worry too much about it. And what we demonstrated recently, depending on which propensity score method you use, it may differ the result. So you can see after all the hard work, just by choosing one particular propensity score matching method, their results can be different. So this is why I have to emphasize the importance of understanding and learning and acknowledging limitation when doing such research.
Okay.
May I ask, how did you get into epidemiology, public health research? I mean, as a kid, were you thinking, well, when I grow up, I will be a public health scientist, or what brought into epidemiology?
Yeah, there's a small article in British Medical Journal about me. So if you read that, you'll get your answer. So getting into medicine was an accident for me. I actually wanted to do maths and engineering, but due to various reasons in my home country, due to war and so on, I didn't put much time in education. And I thought medicine is the easiest thing to do because you just memorize things and just go and churn it out. Whereas maths, you had to spend time thinking. So foolishly chose medicine, as I thought earlier, but now I'm happy I chose medicine. So when I went to medical school, the only thing that excited me was epidemiology and statistics. Even though then I trained in hospital medicine and I did membership for the Royal College of Physicians, I finished my MRCP, etc. In my mind, I always wanted to be an epidemiologist because it felt like being Sherlock Holmes. You're investigating, trying to find the truth. And just like Sherlock Holmes does it by excluding all possibilities, we exclude biases and confounders and chance and then say, this is the truth. So I enjoyed that sort of, so I always was looking towards doing it. The second thing I realized is in medicine, the other thing that interested me was diabetes endocrinology and hematology, simply because they run across your body, the hormones. And so it's fun, it's more physiology, which has more sense of excitement and science rather than a typical heart cardiologist, which is very structural disease, as I call it. So I always loved endocrinology, and I chose actually 4 different pathways to do it. One, public health and epidemiology in diabetes and endocrinology, or a general practitioner, or an endocrinologist, or a metabolic medicine, what we call here as, is it biochemistry? I have forgotten, chemical pathologist, as they call it here. Why did I choose public health? It just gave me more time to do research than an endocrinologist because my kids were very tiny. they'll be creeping through my foot after a night, having done a few nights. Those days we do 7 nights non-stop, and I thought I will never be able to do research the way I want to do, doing night on calls and so on. So I took the easy route of doing public health medicine and doing epidemiology. But there are lots of amazing epidemiologists who are endocrinologists. You have seen Navid Sattar, to a general practitioner. Navid Sattar is more chemical pathologist, but Kamlesh Kunti, who is a general practitioner, but who does also do real world evidence. Then excellent endocrinologists who have done epidemiology. There are so many of them as well. So it doesn't matter which route you take, but take a route that you will enjoy and find your way accordingly.
Thank you.
Thank you, Chris.
Is that time for me to go and have some beers with my colleagues who are waiting after an interview? Not for recording, but yeah.
Yeah, of course, Chris. We are grateful for being here with us and sharing your expertise. I'm sure that you will be willing to clarify any further issues. It's something, or someone has something more to ask you about, general or specific. We would be happy to have you again in the near future in a more specific domain, or specific topic. We will release you now with all our many thanks for you for accepting us our invitation. So, Chris,
thank you, and thank you for the invitation. It's been a pleasure, and hopefully I get to meet some of the early career researchers at some point during the next few years. Thank you.